Okay, so it's 9 o'clock. Hello everyone. Welcome to our today's Museum seminar. It's absolutely my great pleasure to welcome our today's speaker, Dr. Marco Colonna from WashU. I think a lot of us in the audience included very familiar with Marco's CV and then his contribution in science, but just a very brief introduction. Marco currently is endowed professor of pathology and immunology in Washington University in St. Louis. And Marco was born in Italy and then received his medical degree and a specialization in internal medicine in Italy. After he finished the training in Europe, and then he continued to pursue his postdoc training in basic science at Harvard Medical School. And then he became a scientific member of the Basel Institution for Immunology in Switzerland. And then from 2001, Marco has been a professor of pathology and immunology at WashU. And since 2019, Marco was elected as a member of National Academy of Science. Marco's mainly, his research is mainly focused on immunoreceptors. In this field, his lab really contributes a lot, which include identification and the characterization of the killer cell, Ig-like receptors and HLA-C polymorphisms as their inhibitory ligands, as well as the discovery of the LILR and TREM inhibitory and activating receptor families, which I think we're going to hear a little bit today. And then through the analysis of the cellular distribution of the receptors, Marco identify plastomic dendritic cells as source of interferon in antiviral responses and innate lymphoid cells that produce IL-22 in mucosa. His current area of research interested include innate lymphoid cells in mucosal immunity, plus cytoic dendritic cells in host defense and autoimmunity, and then TREM2 and the innate receptors in Alzheimer's disease and cancer. So as I just mentioned, Marco actually did a really lot of groundbreaking discovery and contribute significantly to the immunology science field. And then with that, he was awarded in numerous honors, which include, he was elected as an American Science of Clinical Investigator. And then he's also a member of the Harry Curl Society. And then in 2019, he was elected as a member of National Academy of Science. One thing I want to emphasize is that when I actually look at Marco's CV, it's really unbelievable. He has 321 pre-peer publications and 122 review papers. I have no idea how Marco managed to publish these papers, but he did. It's really extraordinary. So I think without further ado, it's absolutely my pleasure. Marco, really happy to have you on our platform. Please go ahead.
Chuan, thank you very much for the introduction. I'm really flattered. My mom will be proud of me after your introduction.
I'm sure.
So thank you very much for having me. It's really a pleasure to having the opportunity to give this talk. So what I'd like to do today is to review our work on neurodegeneration and cancer and the role of innate immune receptors in this context. And I'll try to convince you that some of these receptors may be valuable for potential therapeutics in these diseases. So these are my disclosures. And we'll start by talking about neurodegeneration, particularly Alzheimer's disease. As many of you know, Alzheimer's disease is a very common form of dementia. The major risk factor is age. And with the aging of the population, of course, this has become a major medical problem. Now, the culprit of the disease is the pathogenic pathway or processing of the amyloid precursor protein, or APP, which is processed by BACE1 and gamma secretase to generate peptides, A beta peptides, A beta 40, A beta 42, which are prone to aggregation and form amyloid plaques. So according to the amyloid hypothesis, the first event in Alzheimer's disease is the generation of these extracellular amyloid plaques, which induce neuronal stress. Stressed neurons then hyperphosphorylate and aggregate an intracellular protein called tau, which is associated with microtubules, and tau precipitates inside the cells to form these neurofibrillary tangles, which cause neuronal cell death, cognitive impairment, and ultimately death. Now, this theory has been supported by genetic studies on familiar Alzheimer's disease or early onset Alzheimer's disease, which have identified mutation in APP and mutation in gamma secretase that facilitate the generation of A beta peptides prone to aggregation. However, since 1992, genetic studies have identified a number of different risk factors for Alzheimer's disease. And interestingly, these risk factors, starting with ApoE in 1992, do not affect the APP processing pathway, but many of these risk factors are actually highly expressed in microglia. Suggesting that microglia is indeed has an important role in the pathogenesis of the disease. So I'm sure that you're familiar with microglia. I just want to remind you that microglia are the brain-resident macrophages. They have this unique ramified morphology, which facilitates interaction with neurons, astrocytes, and oligodendrocytes. And they also have a very unique developmental pathway. They develop extra embryonally from the yolk sac. They colonize the brain very early on, the developing brain. Once the brain development is completed, then microglia will continue to proliferate, to populate the brain with very little input from bone marrow-derived macrophages. Indeed, when we look at the brain with Alzheimer's disease, we see that plaques, amyloid plaques that you can see here in blue, are surrounded by microglia. They send the processes to the plaques, proliferate around the plaques, and try to limit the spreading of plaques, reducing the progression of the disease. So we became interested in this problem because one of the risk factors for Alzheimer's disease is the polymorphism, the R47H polymorphism of a molecule called TREM2 that my lab cloned more than 20 years ago, which is expressed in microglia. TREM2 is one of the members of the TREM family that we clone, which is present both in human and in mouse, although as you can see, the number of genes and their structure is slightly different. TREM2 specifically is a member of the immunoglobulin superfamily, is expressed in microglia, but also in tissue macrophages. It binds to phospholipids and binds to cells that express phospholipids, expose phospholipids, for example, apoptotic cells. It also binds to lipoprotein, for example, ApoE, but also LDL, HDL. TREM2 signals through an adapter called DAP12 that contains tyrosine-based activation motifs. And we and others have shown that DAP12 recruits this protein tyrosine kinase, SYK, and then SYK activates PI3 kinase and PLC gamma 2. And one important point that I'd like to make here is that the signaling pathway is very similar to the signaling pathway of the CSF1 receptor, a receptor that is essential for survival, development, maintenance, and proliferation of microglia. And indeed, we have shown that there's a synergy between signaling of TREM2 DAP12 and signaling of CSF1 receptor. So when we realized that there was this association between TREM2 polymorphism and risk to develop Alzheimer's disease, we wanted to see whether TREM2 has an impact on microglia response to amyloid plaques. And so we used a mouse model of amyloid plaques called 5-XFAD, which contains mutation in human APP and mutation in gamma secretase that facilitate the generation of A beta plaques. And what you can see in the brain of these mice is the accumulation of these plaques that you can see here in green, completely clustered by microglia. But when we look at TREM2 deficient mice, we saw the microglia was unable to cluster around the plaques. And when microglia there's no cluster around the plaques, then plaques are much more diffuse, and they spread their filaments, their fibrils, into the brain parenchyma, and they damage the parenchyma. And in fact, when we look at the axons around the plaques, you can see that there's some axonal swelling in a 5-XFAD mice, in green here, around the plaques, but there's a very strong accumulation of axonal swelling around the plaques in a TREM2 deficient mice. In that I'm not going to show you, we also found that the same microglia defect, the same reduction of plaque density, and the same dystrophy of axons also occurs in a 5-XFAD mice that carry the R47H variant of TREM2, the variant that is associated with Alzheimer's disease. We also look at the impact of TREM2 deficiency on the transcriptome of microglia. The group of Ido Ahmed, together with us, has shown that microglia, during accumulation of A-beta plaques, undergo a progressive differentiation from homeostatic microglia to activated microglia. And we distinguish 4 different branches of activation. One branch is called the disease-associated microglia. These microglia express genes involved in phagocytosis, lysosomal activity, lipid metabolism, adhesion, and tissue repair. Another branch of microglia expresses molecules involved in proliferation. Another expresses very high levels of MHC class II. And the final branch of microglia activation expresses genes induced by type 1 interferon. So we look at these 4 branches of activation microglia in TREM2 deficient mice. Here you can see the 4 different trajectories of microglia activation: MHC class II, interferon responsive, proliferating microglia, and DAM. And you can see that in mice that express human TREM2 at normal level, that all these different trajectories are equally represented. But when we look in mice that have a defect of TREM2, or mice that carry the R47H mutation of TREM2, we see a very strong reduction of all these trajectories. So in other words, TREM2 is important to sustain transcriptional activation of microglia. And so we asked what is the mechanism, and this was a problem that was addressed by Shutang Wang in a lab, who is now a professor at University of Hong Kong. As I mentioned, TREM2 and APP12 recruit SYK, and we know that SYK is important to activate multiple signaling pathways. So we also noticed that in 5-XFAD mice, SYK is phosphorylated in microglia, whereas we see very little phosphorylation in wild-type mice, suggesting that SYK is activated during accumulation of A-beta plaques. And so we generated a mouse that lacks SYK specifically in microglia, and then we crossed these mice with the 5-XFAD model of Alzheimer's disease. And so we saw exactly the same phenotype that we saw in TREM2 deficient mice. Again, we see that microglia is unable to form clusters around the plaques. The plaques are bigger, they spread out, they diffuse the disease. And indeed, when we look at the accumulation of A-beta peptide, of A-beta plaques, we see much more accumulation of A-beta plaques in a SYK deficient mice compared to a wild-type mice. And in data that I'm not going to show you in the interest of time, we also shown that in the absence of SYK in microglia, mice have behavioral defects, particularly learning defects. We also look at the pathway downstream of SYK, and we're particularly interested in a PI3 kinase AKT pathway, because this pathway is important to control JSK3 beta, which inhibits the proliferation of cells. And this pathway is also important to phosphorylate and activate the mTOR pathway that sustains cell growth, cell proliferation, and protein synthesis. And indeed, when we look at microglia, in mice that lack SYK and accumulate A-beta plaques, we saw a defect of a phospho-AKT phosphorylation. We saw a defect of phosphorylation of JSK3 beta. And we also saw a defect of 2 genes, NDRG1 and S6 kinase, which are phosphorylated by the mTOR pathway. So based on these results, we postulate that during neurodegeneration, particularly during Alzheimer's disease, the brain contains a number of molecular patterns can activate microglia. For example, protein aggregates, prototoxic proteins, apoptotic neurons, damaged DNA, damaged myelin. And these receptors will activate, and this molecule will activate different receptors. For example, scavenger receptor, TREM receptor, perhaps toll-like receptor, node-like receptor. And this will lead to microglia activation and generation of 4 different trajectories of microglia activation. However, this activation requires the TREM2 DAP12-SIG pathway to activate mTOR and to sustain activation. In the absence of this pathway, microglia is not fit and therefore is unable to sustain responses to A beta plaques. Now, this idea led us to possibility that we can activate TREM2 in microglia to have a beneficial effect in amyloid pathology. And this is working we done in collaboration with different companies. And in just in the interest of time, I'll just show you one example. Here we generated transgenic mice that express the human common variant of TREM2 or the R47H variant of TREM2. We took these transgenic mice, crossed them onto TREM2 deficient mice to eliminate endogenous TREM2, and then cross them onto 5-XFAD mice. And then we let these mice accumulate plaques until 5 months of age. At this time, we started injection of high doses of the antibody intraperitoneally once a week for 3 months. And then at the end of the treatment, we look at the pathology of these mice. And this is just one result that we obtained. Here you see the brain section of mice that have been treated with a control antibody. In blue, you see the plaques, and in red, you see the axonal swelling around the plaque. So you see a significant damage of axons around the plaques. But when you look at mice that have been treated with anti-TREM2 antibody, agonistic antibody, we see a reduction of the damage around the plaques, which is statistically significant. And in data that I'm not going to show you, but we publish it, we also show that anti-TREM2 ameliorates the behavioral traits of these mice. And a version of this antibody now has completed phase 1 clinical trial and is currently in phase 2 clinical trial for Alzheimer's disease. So based on this result, we postulate that because TREM2 and SIG are expressed in human microglia, the therapeutic activation of microglia in Alzheimer's disease can be achieved by stimulating TREM2 with monoclonal antibodies, with small molecules, or also by stimulating other receptors that activate the SIG signaling path. Now, what I show you so far is that we can activate microglia by engaging TREM and activating receptor that activates SIG. However, the activation of microglia is actually controlled by a balance between activating receptor and inhibitor receptor. Activating receptor activate what we call the ITAM pathway that leads to phosphorylation activation of SIG. The inhibitor receptor contain long cytoplasmic tail, which contain tyrosine-based inhibitory motifs. And this motif records protein tyrosine phosphatase, SHIP1 and SHIP2. And these phosphatases inhibit and dephosphorylase what has been phosphorylated by SIG. So the activation of microglia is actually controlled by a balance between inhibitor receptor and activating receptor. And we're particularly interested in this family of receptor called LILRs. This family of receptor actually was cloned by my laboratory more than 20 years ago and includes 5 different inhibitor receptors. You have B1, 2, 3, 4, and 5. They all have cytoplasmic tail contains ITIM, and they all recruit SHIP1 and SHIP2. The mouse homolog of these receptors is TRB. These receptors are encoded by 2 gene clusters on human chromosome 19, which are close to the killer cell Ig-like receptor. We were interested in this receptor because this region has also been linked to risk all Alzheimer's disease, although the susceptibility to Alzheimer's disease is less penetrant than ApoE or TREM2. And we're particularly interested in this receptor called LILRB4, which has two immunoglobulin domains. LILRB4 is expressed in monocytes, in dendritic cells, in macrophages, in plasmacytoid DC, and also in B cells. We're interested in this receptor because a group of Alex Cheng at UT Southwestern has shown that LILRB4 binds to ApoE in a context of tumors. And the ApoE, of course, is a major risk factor for Alzheimer's disease. We also found, in collaboration with NGM Biopharmaceutical, that LILRB4 binds to fibronectin. And recent work at Columbia University have shown that one variant of fibronectin 1 is protecting against Alzheimer's disease in subjects that are carrying the ApoE4 allele, which gives susceptibility to Alzheimer's disease. So LILRB4 binds to two molecules, ApoE and fibronectin 1, which are both related to Alzheimer's disease. And so the question we ask is whether we can harness LILRB4 for treatment of Alzheimer's disease. And this work was done by Jin-Chao Hu and Yun Chen in the lab. So first of all, we noticed that we look at single nuclear RNA-seq databases that we generated. This is a microglia cluster, and we found that LILRB4 is highly expressed in microglia. Additionally, LILRB4 is increased during Alzheimer's disease. In fact, when we look at the all LIL receptor, LILRB4 is the only one that is upregulated during Alzheimer's disease. And when we look at brain section, this is a brain section that I showed you before. We see again the plaque, we see the microglia around the plaque. And when we stay microglia for LILRB4, we see that LILRB4 is highly expressed in microglia around the plaques, as you can see here. Also, I'd like you to notice that plaques also contains ApoE. Most of this ApoE is non-lipidated, but we have shown, and Alex Chen has shown as well, that LILRB4 can bind to both lipidated and non-lipidated ApoE. So we postulate that microglia around the plaques expresses LILRB4, engages with ApoE, and receives an inhibitory signal that blocks activation of microglia. So to test this hypothesis, we generated a transgenic mouse, a back transgenic mouse that expresses a whole telomeric cluster of LILR, which contains LILRB4. And then we cross this mice onto the 5-XFAD mice to look at the impact of LILRB4 on accumulation of beta plaques. So this is our transgenic mouse. You can see the plaques. You can see in red microglia around the plaques. And in green, you can see the LILRB4 expression in microglia around the plaques. You see very high expression. So we have recapitulated expression of human LILRB4 in microglia around amyloid plaques. And so now we ask whether we can impact the beta accumulation by using a monoclonal antibody anti-LILRB4. So as we did in a previous experiment, we take our transgenic mice, we let them accumulate A beta plaques, and then we treat them with high doses of anti-LILRB4 monoclonal antibodies once a week for 8 weeks, and then we look at the accumulation of A beta plaques. Here we look at the accumulation of antibody in the brain, and you can see that we reach significant amount of antibody in the brain, although we inject the antibody intraperitoneally. So that's exactly what happened. We see a very strong reduction of A beta plaques in mice treated with anti-LILRB4 antibody. Here we check the accumulation of A beta plaques with methoxyl-4 or with the antibody 6010. And in both cases, we see a clear reduction of accumulation of A beta in these mice. And in data that I didn't show you, we actually demonstrated that treatment to anti-LILRB4 is increasing phagocytosis of A beta by microglia. The question we ask then is, we know that LILR4 binds human ApoE, but does LILR4 binds mouse ApoE in vivo in our model? And so to address this question, we generated a reporter cell line in which we convert the inhibitor LILR4 into an activating receptor by making a chimera that expresses the extracellular region of LILR4 and the intracellular region of CD3 zeta. Now, if we engage LILR4 with a ligand, for example, ApoE, we will trigger a signaling molecule, a signaling pathway that will induce the expression of GFP. And so when we take this reporter cell line and we engage with ApoE, we see indeed that the reporter is activated. And the activation of the reporter is by ApoE, by mouse ApoE, is dose dependent. Importantly, when we perform this experiment in the presence of the anti-LILR4 antibody, we completely block the activation of the reporter, suggesting that the anti-LILR4 antibody that we use has an antagonistic activity. And so what I've shown you is that human LILR4 is expressed in microglia around the plaques and correlates with amyloid pathology, that we can reduce the A beta burden in our mouse model using anti-LILR4 antibody. And we've shown that anti-LILR4 antibody can increase the phagocytic activity capacity of microglia. And we think that the antibody actually blocks the interaction between LILR4 and ApoE in the A beta plaque, releasing microglia from inhibition and resulting in activation of microglia. So, in conclusion, we think that therapeutic activation of microglia in Alzheimer's disease and other neurodegenerative diseases can be obtained not only by using antibody anti-TREM2, but also by using antibodies, small molecules that block LILR4 or block other inhibitor receptors that are very important in inhibiting microglia activation. So let's go back now to TREM2. And since we started to work on TREM2 in neurodegeneration, TREM2 has become a very popular molecule, as I'm sure many of you know. In fact, TREM2 is expressed in many tissue macrophages and has been found in adipose tissue, in the liver, and in vessels. And it has been shown to have several effects. For example, in obesity, TREM2 is highly expressed in lipid macrophage, in adipose tissue macrophages, and has some effect on lipid metabolism. TREM2 is highly expressed in liver macrophages during fatty liver disorder, is expressed in foamy macrophages in atherosclerosis. But we were particularly interested in cancer because TREM2 is expressed in tumor-associated macrophages. Indeed, we found that we tested many different human tumors. Here you see section of many different human tumors: skin, liver, lung, bladder, breast, stomach, glioma, cutaneous melanoma. And we find very strong expression of TREM2 in macrophages that infiltrate the tumor. We also noticed in a TCGA database that high level of expression of TREM2 in two types of cancer, colorectal carcinoma and triple-negative breast cancer, is associated with low survival of a patient, suggesting that TREM2 has expression in tumor-associated macrophages and has a detrimental effect. Now, we know that tumor-associated macrophages have immunosuppressing activity. They reduce T-cell activation through a number of different mechanisms, through inhibitory ligands, through secretion of anti-inflammatory cytokines, through secretion of metabolites, and through secretion of prostaglandin. And so the question we ask is whether we can harness TREM2 signaling, we can block TREM2 signaling, and reprogram tumor-associated macrophages from immunosuppression to immunoactivation. And this was work that was done by Martina Molgora, together with Jin-Chao Hu and Yun Chen. So in the first set of experiments, we took 3 different tumors models, and we inject it subcutaneously in wild-type and TREM2 knockout mice. And we noticed that this tumor grow less in TREM2 knockout mice. The reduction of tumor growth was not dramatic, but was very consistent with different tumor models. We isolated macrophages from these tumor models, specifically here from the MCA carcinoma, subcutaneous sarcoma, and we look at macrophages by single-cell RNA-seq. You can see that macrophages express TREM2, perhaps at different levels, and we can see different cluster of macrophages. And when we look at TREM2 deficient mice, we see that there's a reduction of these macrophages, CX3, CR1 macrophages. They're proliferating. They're also cycling. We also see a reduction of this population of macrophages, CD206, that have been associated with immunosuppression. On the other hand, we see an increase of this population of macrophages that expresses CXCL9 and CD83. These changes in the macrophage landscape are associated with an increased presence of T cells, CD8 T cells, that express interferon gamma and express PD-1. And we confirm this also by flow cytometry. As I mentioned, the effect was very consistent, but was not dramatic. And so we thought that perhaps we can amplify the effect of TREM2 deficiency by associating TREM2 deficiency with immune checkpoint blockade, and particularly anti-PD-1. So to test this hypothesis, we used this model, this MCA sarcoma, that was developed in our institution by Bob Schreiber. Now, this tumor model is actually very susceptible to checkpoint blockade. In fact, if we inject anti-PD-1 very early on, you see the tumor is completely rejected. So to mimic resistance to anti-PD-1, we use a suboptimal scheme of therapy. We injected the anti-PD-1 very late. And if we do this, then the tumor is not rejected, but it's simply dormant. It doesn't grow anymore. And so we performed this experiment in TREM2 knockout mice. So as I show you, this MCA sarcoma grows in wild-type mice. It grows less in TREM2 deficient mice. It becomes dormant in mice treated with suboptimal anti-PD-1, and is completely eliminated when suboptimal anti-PD-1 is performed in TREM2 deficient mice. And we obtain similar data with the MC38 colorectal carcinoma model. Now, we wanted to know whether we can obtain the same effect with an anti-mouse TREM2 antagonistic antibody. And so indeed, here we use the anti-TREM2 monoclonal antibody. You see that the monoclonal antibody alone is sufficient to reduce tumor growth. When we use anti-PD-1 alone, we induce tumor dormancy. But when we use together anti-TREM2 plus anti-PD-1, we eliminate the tumor. Now, we isolate macrophages from these tumors, and we look at the impact of the different tumor treatment. Now, first of all, again, you can see that all macrophages in the tumor express TREM2, perhaps at a different level. And when we look at macrophage in mice that have been treated with anti-TREM2 plus anti-PD-1, we see a reduction of this population of macrophages that expresses CD206. We also see a reduction of macrophages that are proliferating. On the other hand, we see an expansion of macrophages that express the inducible nitric oxide synthase. This population has been associated frequently with an immunostimulatory function of macrophages. And these changes in macrophages are also associated in changes in T cells. We see an increase of CD8 T cells that produce interferon gamma, and we also see an increase of CD4 T cells that produce TNF. And so what I've shown you is that in TREM2 is expressed in tumor-associated macrophages. It's highly expressed in tumor-associated macrophages and is associated with reduced survival in individuals with triple-negative breast cancer and colorectal carcinoma. I've shown you that TREM2 deficiency or blockade of TREM2 with a monoclonal antibody has some protective effect in different tumor models and synergizes with anti-PD-1, at least in one model. And of course, these changes induced by anti-TREM2 treatment induce a change in myeloid compartments and especially in T-cell activation. And so, based on this result, we speculate that TREM2 blockade may be beneficial in patients that are not responsive or hyperresponsive to checkpoint blockade. And I like to make the point that this result is being reproduced now by different groups, particularly by Ido Amit, by Max Connell, by Miriam Merad, and by Eleonora Timper. Now, in the final minutes of my talk, I'd like to tell you some new data that have been very recently published, I think will be published this week, in which we investigated the mechanism by which TREM2 deficiency improves anti-PD-1. And this work was done by Blanda Di Lucia together with Martina Molgore and Daria Kantakova. And this work was done in collaboration with Jeff Gordon. In the initial experiment that we performed, we took wild-type mice and TREM2 deficient mice that are co-housed from birth to attenuate every difference in a microbiota. We inject them with a tumor, and then we separate the two different genotypes, and then inject them with anti-PD-1. And after 20 days, we look at the tumor growth. And as I mentioned, TREM2 deficient mice, we see reduced growth. Mice treated with anti-PD-1, we see tumor dormancy. TREM2 deficient mice treated with anti-PD-1 completely reject the tumor. But we also done a second experiment in which we take wild type and TREM2 deficient mice that be housed from birth. We inject them with a tumor, and then we maintain them in the same cage after anti-PD-1 treatment, and then we look. So if we do this, again, TREM2 deficiency reduces tumor growth, but now when we associate anti-PD-1 plus TREM2 deficiency, we no longer see synergy between the genotype and the treatment. So this, of course, suggested that the microbiota has importance in the TREM2 effect on anti-PD-1. And to test this hypothesis, we did a sickle microbiota transplantation experiment. And this is a scheme of the experiment that we did. We take, again, wild-type and TREM2 deficient mice. We inject them with a tumor. We separate them. And then we take sickle content before treatment anti-PD-1 at day 0, or after treatment anti-PD-1. And we take the sickle content for fecal transplantation. So now we take wild-type and TREM2 deficient mice. We treat them with antibiotics, and then we reconstitute them with sickle content from TREM2 deficient mice or TREM2 wild-type mice at day 0 prior to injection of PD-1, or at day 12 after injection of PD-1. After we reconstitute the microbiota of these mice, we inject them with a tumor. Then we separate the two genotypes, and then we treat them with anti-PD-1, and then we look at the tumor growth. So this is just a summary of the experiment. Here we take wild-type mice and reconstitute them with sickle content of knockout mice at day 0 prior to treatment with anti-PD-1. You see the wild-type mice do not reject the tumor, even if we reconstitute them with the sickle content of knockout mice. However, when we take wild-type mice and we reconstitute them with the sickle content of knockout mice at day 12 after injection of anti-PD-1, we see that the wild-type mice now reject the tumor. Conversely, here we take knockout mice that normally reject the tumor. We reconstitute them with the sickle content of wild-type mice at day 0 prior to injection of anti-PD-1. The knockout mice reject the tumor. But now we take knockout mice, and we reconstitute them with sickle content of wild-type mice at day 12 after injection anti-PD-1. And now knockout mice no longer reject the tumor. We also perform some antibiotic experiment. As I mentioned, TREM2 deficient mice treated with anti-PD-1 reject the tumor. TREM2 wild-type mice treated with anti-PD-1 do not reject the tumor. However, if we treat both groups with ampicillin, they do not reject the tumor. If we treat them with vancomycin, we see no rejection of the tumor. If we treat them with metronidazole, the TREM2 deficient mice still reject the tumor. So this suggested to us that the bacteria that are responsible for the rejection of the tumor are gram-positive bacteria. Now, we did several experiments here. We identified the species by cysteine sRNA, ribosomal RNA-seq in different groups of mice. I'm not going into details here. We look at TREM2 wild type and TREM2 deficient mice, separated, non-separated, cohoused, non-cohoused, treated with antibiotics. And the point I like to make is that at the end of this complicated experiment, we end up with one single species of bacteria that is associated with the rejection of a tumor in TREM2 deficient mice. And this species is Ruminococcus nievus. So why do we see expansion of Ruminococcus nievus in TREM2 deficient mice treated with anti-PD-1? We know from work of gastroenterologists that Ruminococcus nievus is actually expressed in patients with Crohn's disease and is particularly expanded during inflammatory flares. We also know from the work of John Clardy and Ramesh Gabier that Ruminococcus nievus contains a capsule of polysaccharide that stimulates TLR4 and is inducing inflammatory responses. So we speculate that perhaps we're inducing some gut inflammation in anti-TREM2 knockout mice, in TREM2 deficient mice, and this facilitates the expansion of our nievus. In fact, we didn't see any clinical colitis, obvious colitis. We didn't see any changes in histology. We didn't see any fecal lipocalin. However, we did see that in TREM2 knockout mice, anti-PD-1 is inducing increased production of inflammatory cytokines like IL-17 and TNF-alpha, and reduces the production of IL-10. So we think that anti-PD-1 in TREM2 deficient mice is inducing a subclinical inflammation. And how is that possible? Well, we know that TREM2 is expressed in macrophages. And so we postulated that TREM2 perhaps is also expressed in intestinal macrophages. And TREM2 deficiency facilitates colon inflammation. So to address this hypothesis, we take TREM2 wild type and TREM2 deficient mice. We treat them with tumor. We treat them with anti-PD-1. And then we isolate the gut of these mice after injection of anti-PD-1, and we perform single-cell RNA-seq of intestinal macrophages. So these are the macrophages we isolate. As you see, all macrophages express TREM2. And now we find different clusters of macrophages, and we look at these clusters in different treatment. These are knockout mice. These are knockout mice treating anti-PD-1. These are the wild-type mice, and these are wild-type mice treating anti-PD-1. And what we see is that three clusters, cluster 0, 1, and 2, change in a knockout mice treating anti-PD-1. Cluster 0 and cluster 2 are increased. Cluster 1 is decreased. So when we look at cluster 0, we see cluster 0, which is increased, expresses pro-inflammatory cytokine, CCL3 and CCL4. CCL2, cluster 2, which is also increased, expresses interleukin-1 beta, NLRP3, CD80. These are markers of inflammation. Cluster 1 that is decreased expresses CD163, MRC1, or CD206, and stabilin-1, which are markers of immunosuppression. So in other words, treatment of anti-PD-1 in TREM2 deficient mice is inducing pro-inflammatory function, and inflammatory function bias on colony macrophages, facilitating pro-inflammatory phenotypes. And so what we think is happening here is that when we treat mice with anti-PD-1 in a gut, nothing happens in wild-type mice. But in TREM2 deficient mice, treatment with anti-PD-1 induces a pro-inflammatory bias of intestinal macrophages, and this facilitates the expansion of our neighbors. And of course, our neighbors then facilitate the anti-tumor T-cell response and reduces tumor growth. So finally, we want to demonstrate whether our neighbors can replace TREM2 deficiency in enhancing anti-PD-1. And so this experiment that we did, we take wild-type mice, we inject them with a tumor, we gavage them with our neighbors, and then we treat anti-PD-1. And after 20 days, we look at tumor growth. So as you can see, mice treated anti-PD-1 alone, suboptimal anti-PD-1, do not reject the tumor. But mice that have been gavaged with Romiococcus nevus, they reject the tumor, even if there's no TREM2 deficiency. We done a control experiment with another obligate anaerobic bacteria, Clostridium shingles, and we see no effect. So the effect is specific of Romiococcus nevus. We wanted to demonstrate that our nevus plus anti-PD-1 has an impact on immune infiltrate of the tumor. And so what we did here was to repeat the same experiment in which we take wild-type mice, inject them with a tumor, gavage them with our nevus, treat an anti-PD-1, and then isolate CD45 positive cells from the tumor and look at the impact of our nevus on this infiltrate. When we look at T cells, we see an expansion of effector T cells, both CD4 and CD8. But when we look at macrophages, we also see an expansion of immunostimulatory macrophages and a reduction of immunosuppressive macrophages. Specifically, when we look at the signatures of macrophages, we see that our nevus is inducing a reduction of cluster of macrophages express TGF-beta receptor 1. So our nevus is reducing responsiveness of the tumor to TGF-beta. On the other hand, we see an increase of macrophage cluster that express interferon gamma receptor 1. So our nevus is increasing responsiveness of tumor macrophages to interferon gamma. And so to conclude this part of the talk, I've shown you that the housing condition facilitate the synergy between anti-PD-1 and TREM2 deficiency. And therefore, microbiome from TREM2 deficient mice is what is important to synergize with anti-PD-1 to facilitate tumor rejection. And indeed, anti-PD-1 in the absence of TREM2 induces the expansion of this bacteria called R. nevus, and R. nevus alone is sufficient to synergize with anti-PD-1 to induce tumor rejection without TREM2 deficiency. So, of course, there's a number of different questions that we're trying to address now. First of all, we want to know why intestinal inflammation, subclinical intestinal inflammation, is facilitating the expansion of R. nevus. We can think about different ideas. One idea is that inflammation can induce a degrees of hypoxia, and hypoxia, of course, can facilitate the expansion of anaerobic bacteria like R. nevus. But the most important question is that how does R. nevus facilitate T-cell responses to tumor at a distant site? We can think about different possibilities. We can think that R. nevus can facilitate T-cell migration from the gut to a tumor. Another hypothesis is that perhaps R. nevus can leak through the gut and reach the tumor bed and facilitate tumor rejection in the tumor. Another hypothesis is that R. nevus produces bacterial metabolites that promotes immune activation. These are all questions that we're trying to address currently. I think I mentioned all the people that did this work. I just like to acknowledge this is my lab. It's a large lab, but they have several people leaving this year. So if anybody's interested, please let me know. And I'd like to make some acknowledgement. I'd like to acknowledge David Holtzman and John Kipnis for help in studying neurodegeneration, Bob Schreiber for tumors, Jeff Gordon for our work on microbiota. We have important collaboration with the Weizmann Institute for microglia. We have important collaboration also with different brain research institute that provide us with brain samples from human Alzheimer's disease. We have industrial collaboration. And finally, I'd like to acknowledge also our collaboration with Ramit Xavier at the Broad Institute. So I think I will stop here, and I think we have time for questions if there's any question. So thank you for your attention.